Hello everyone and welcome to our talk on FlyBrainLab, an interactive open computing platform for exploring the Drosophila brain. My name is Mehmet Kerem Türkçe. I am a PhD candidate at BioNetLab in the Electrical Engineering Department at Columbia University. I am presenting this work on behalf of my research advisor Ora Lazar and my lab mates Tinkai Liu and Yin Zhao. We are grateful to have this unique opportunity to share with you our work on exploring, modeling, and comparing models of the brain of Drosophila. Without further ado, let's begin. You might be asking right now, what is Drosophila? Drosophila, or specifically the species Drosophila melanogaster that we study, are well-known common pests in households that are attracted to rotten fruits into which they lay their eggs. This has led to their popular name, the common fruit fly. Drosophila is a popular model organism in research, and historically 5 Nobel Prizes have been awarded to a total of 9 scientists for their research based on the fruit fly. The long-lasting popularity of the fruit fly has led to a huge wealth of data on the structure of its brain, its behavior, and its genetics, rendering it a good subject for neuroscience research. To be more specific, for Drosophila, we have an immense amount of data on its brain throughout different stages of development, which allows us to pinpoint lineages of different neurons. We have a great wealth of data on the brain regions, or neuropils that make up the brain and how they are connected. We know a lot about how different cells can be classified into types based on their morphology, primary neurotransmitters, function, or genetics. In addition to having the capability to record the activity of neurons, we also know about the behavioral repertoire of Drosophila and the groups of neurons associated with these behaviors. Finally, in the last few years, data on the neural circuits in the brain at single synapse scale is now starting to become available, which gives us neurons and how they are connected, allowing us to explore neural circuits in unprecedented levels of detail and form hypotheses about their function, especially when coupled with the rest of this data. We expect the whole Drosophila brain to be available in the next few years. In the meantime, we work with 3 specific datasets in particular. The first dataset we work with, and the first dataset we historically started to work with, is the so-called fly circuit dataset, the first version of which was published in 2011. Fly circuit dataset is composed of about 20,000 randomly sampled neurons from different specimens registered to a common brain template. For many years, it has been central to researchers that study information flow between fly brain regions, but it fails to support further advances in understanding the functional logic of neural circuits for several reasons. Number 1, the uniqueness of each neuron and the completeness of neurons within a neuron type could not be established because each neuron is randomly sampled from a different individual. Number 2, the dataset does not have synaptic connections, which means we don't know how neurons are connected to one another. Each of the thin lines you see here is part of the so-called skeleton of a single neuron. And in this custom render, we have thousands of the fly circuit neurons present. At the center is the so-called central brain, and the 2 parts to the side are the visual system. The second dataset we worked with was released in January this year, and it took us a week after its release to integrate the dataset into our platform. The HemiBrain dataset, the extent of which I am showing only approximately here to give an idea of its size, is a high-resolution dataset that contains more than 20,000 traced neurons and synapses between them. The quality of these tracings, as well as the annotation of the cell types provided by the maintainers, has greatly extended our knowledge about how neural circuits in the central brain are wired. Finally, a collaborative project has been going on to image and trace the neurons and their synaptic connections in the brain of larval Drosophila over the last few years. Due to the similarity of certain structures in the brain of larval Drosophila to the adult, as well as its capability for a range of interesting behaviors, Drosophila larva is popular. We are interested in using this data for comparative studies and for understanding common capabilities of the adult and the larva. As an illustration of what we do with this data, let me present an example about the early visual system of Drosophila. Drosophila have a compound eye structure composed of about 750 so-called ommatidia. If we were to focus on what is going on right within one of these ommatidia, we would observe a structure like this, in which different neurons are colored with different colors. These are the neural circuits whose computational structure we are interested in. For us, these are circuits not composed of transistors, but rather of neurons and synapses. These executable circuits can be interrogated to understand how neural circuits function. Historically, there has been a lack of structural data on the wiring of these circuits, and with the data sets that I was describing just a moment ago, this limitation is no longer there. However, all of this structural data today is stored in different formats that are difficult to parse and sometimes require heavy domain expertise to process and format into neural circuits like what I'm showing here in the second row. Now, I would like to explain our main interest in all of this data, their integration and circuit construction. As computational neuroscientists, our main goal is to understand brains. To understand brains, we have to understand how neural circuits perform computation. To do so, we want to use the information we have on these circuits rather than black box approaches to constrain our models appropriately and achieve better correspondences in order to discover the computational features of these circuits that generalize beyond the experiments. In this quest, it is of value to know what these circuits actually look like. Now, our discussion so far perhaps masks the greatest challenge facing us as computational researchers, and that is the lack of a central ecosystem that brings all of this data together, gives people access to them from a Python kernel with consistent data formats, integrates this data with simulations to make the neurons modeled transparent so that the features of the models can be compared against the neural circuits to judge their hidden assumptions. FlyBrainLab provides, within a single working environment, 3D visualization and exploration of connectome data, interactive exploration of executable circuit diagrams, and real-time code execution. This brings us to what FlyBrainLab looks like. I will describe FlyBrainLab by starting with our front end, which we call NeuroMinerva. Now, again, here is our concept of what we seek to accomplish, and here is how we implemented it. This is the user interface that we use for our research on neural circuit exploration, execution, and comparison. This is NeuroMinerva, the front end of FlyBrainLab built on top of JupyterLab. On the screen, you see a couple of widgets for visualizing structural data and computational models for the fruit fly brain. I will go over them one by one, but first, I want to get this out of the way. The visualization widgets we provide are associated with specific Python kernels connected to notebooks, which can be treated as standard notebooks. Every data we visualize can be accessed from the Python kernel or requested via documented Python APIs. And neural circuits you explore can be compiled into executable models that can be customized and run on GPUs using FlyBrainLab. We use a custom simulation library we call NeuroKernel and a neural circuit implementation library called NeuroDriver to implement these models, and the library called NeuroBallad for simplifying circuit specification. JupyterLab's built-in local file access panel is also quite convenient for the computational work that we do. For visualization of neuron morphology and metadata, we have two widgets. The NeuroNLP window allows us to use natural language queries to retrieve data from datasets, and the info panel contains a large variety of data on a neuron, including its partners. Here, I will give an example of the interface in action, showing these two widgets. When you open FlyBrainLab, you will see an option to create an FBL or FlyBrainLab workspace. Clicking on this, you will be prompted to choose the dataset that you would like to connect to by default. Here, I will choose the Fly Circuit dataset, which will load the associated brain regions. You are free to customize your workspace however you want. For this demo, I will maximize the NeuroNLP window. I will then simply write show neurons in the ellipsoid body, where ellipsoid body is a region in the Drosophila brain. The neurons will be retrieved from the backend and their morphology will be visualized. I can rotate my screen. I can hover over neurons to highlight them. But more importantly, I can click on them to actually retrieve information about them. As you can see, this information contains their unique name, their database, the synaptic partners that are inferred using an algorithm that I'm not going into detail here. I can use this list of neurons that they are connected to bring in these partners into my workspace. This is just a simple demo demonstrating how you can construct neural circuits during exploration using NeuroMinerva. Now I would like to describe how we have implemented the capabilities we have just looked at. Now the data that you see come up in FlyBrainLab is data coming from our graph database, NeuroArc, which can be running on the same computer or on a user-designated server. NeuroArc is built on top of OrientDB, and nodes in the graph correspond to neurons, synapses, computational models, neurotransmitters, brain regions, and more. This information is used in queries to return all available information on a neuron, which can be accessed from the info panel or directly as a Python dictionary via using our API. This data contains information such as name of the database the neuron is coming from, its name, its 3D morphology, as you can see to the left, the database reference ID, cell type, its neurotransmitter if it is known, number of synapses it has on each brain region, number of neighboring neurons, and number of synapses between this neuron and that neuron, circuits modeling this neuron, and computational models used to model this neuron in these circuits. And finally, morphometric data such as surface area, volume, etc. The NeuroNLP interface has many more capabilities than just showing something. We can remove neurons matching a certain characteristic or keep only those matching one. Here, I remove GABAergic neurons first, and then choose to keep only the glutamatergic neurons, further filtering what I have in my current workspace. One can also choose to show neurons connecting different brain regions to one another, since as I have shown, we track this information in our database as well. Secondly, we have the NeuroGFX window, which allows us to visualize neural circuits with interactive circuit diagrams. Rather than talking about these capabilities, I will show an example using the same Ommatidium example I have presented earlier. We can click on the lamina region in the circuit diagram of the entire fly brain, which you can see at the bottom half of the screen, to go to the lamina. And double-clicking on what we observe, again, we enter into a single Ommatidium. FlyBrainLab circuit diagrams are programmable, and the circuit diagram automatically finds the neurons whose descriptions exist in this SVG file and brings them to our workspace. We associate a number of elemental operations with these diagrams. Clicking on an active element will disable it and remove the neuron from our simulations. These changes are reflected in the Python kernel and can be used to study the effects of disabling a neuron on a neural circuit. I want to mention that FlyBrainLab was built to enable many different kinds of interactive circuit visualizations based on different circuits, and over time we have created a large variety of scripts for using as a way to visualize circuits. For example, here I am showing a diagram for the whole early visual system of the fly based on the connectomics data, in which additional user-given information allows for neurons to be distributed to different rows based on their neuropils. But this can be customized visually and adapted for different circuits if we wanted to. Secondly, here I am showing the diagram we have created for the entire central complex, a multi-neuropil structure in the Drosophila brain. This was a custom diagram that was designed in Inkscape, for which we use the SVG element properties and the script to add interactivity to the diagram. The example that I was showing earlier is similarly based on a custom SVG file. Lastly, we provide examples for using interactive diagrams from the connectome data retrieved from the NeuroArc database using Python scripts for which we provide standardized scripts enabling interactivity. These three different methods of circuit construction I have mentioned have some common features in terms of circuit interactivity. Clicking on neurons in a diagram allows for adding them to or removing them from the workspace. These changes are reflected in the computational model. Secondly, users can create hierarchical diagrams where nodes do not only correspond to neurons. I will show an example to this later. Finally, it is possible to add the capability to change the model parameters or computational models of individual components from the front end. Now that I have presented the capabilities we provide in the front end, I want to briefly talk about the architecture of FlyBrainLab. So far, I have talked about NeuroMinerva, which forms the user-side front end I am presenting at the top row. The second row I just unveiled shows what we have in the user-side backend. Of these components, the FlyBrainLab client is the one that communicates with our servers. We use the crossbar library to communicate with the server, retrieve data, parse it, and send it to NeuroMinerva. Our server software is also written in Python and contains our database. Commands from the FlyBrainLab client are interpreted, executed, and the results are sent back either at once or in chunks where the data is too large. In practice, we find the neuron morphology requests and simulation result retrieval to be two such cases in which data needs to be retrieved in chunks. Importantly, I want to mention that FlyBrainLab is designed with modularity in mind, supporting users and developers with different technical backgrounds, with remote access to the backend servers anywhere in the world. On the left, I am showing the different proficiencies that are in demand for helping with the development of our platform that those interested in helping computational neuroscience can contribute to. Finally, it is our hope that the accessibility of JupyterLab as an environment makes it easier for newcomers to be introduced to the modern challenges in computational neuroscience via FlyBrainLab. Before moving on to examples showing some sample circuits we have created and simulation results, I want to briefly show the capabilities we enable via a number of utility libraries we have created to better analyze the data retrieved from the database during our exploration. FlyBrainLab features a number of utility libraries that aim to make it easier to work with the data retrieved from the servers. Firstly, there is the NeuroEmbed library, which creates node embeddings for a graph in which neurons are nodes and synapses are edges. On the left, I am showing a bunch of neurons closely connected with one another. On the right, I am showing the output of one of the models implemented in NeuroEmbed, which embeds neurons in a 2D space in which closeness of nodes is highly predictive of their human annotated cell types. We have used hell.dot cell types to see how well our model worked in this scenario. These groups can then be analyzed and interrogated to see how correct human annotations are, or to find new types of cells. Secondly, we have the NeuroGraph library, which allows for the discovery of connectivity patterns by using community detection or clustering algorithms on specified neural circuits. Here I am showing the adjacency matrix of more than 20,000 neurons in the hemibrain dataset, ordered according to communities extracted by applying the Louvain algorithm using Neurograph. We have more libraries for data visualization, querying in Python, and for generating morphometric measurements for individual neurons when that data is available, such as axon length, surface area, or neuron volume. With FlyBrainLab, we seek to provide a unified environment for the development of brain circuit models. For the remaining part of this talk, I will go over 2 case studies of circuits that we have looked at as examples to the capabilities of FlyBrainLab. The subject of our first study will be the central complex, the subcircuits of which have appeared before in this presentation. We have created a highly complex circuit diagram that replicates our observations about the circuitry of the central complex. We can highlight regions by hovering over them, or we can click on regions to ablate entire regions at once corresponding to multiple neurons. Secondly, our diagram, as I'm about to show in a second, is hierarchical, allowing us to enter into regions and observe how groups of neurons are connected to one another in these subcircuits, as you can see. In this quest, we have started with a work that was published in 2017 by former members of our lab. Using a slightly different and interesting input stimulus, now with an additional but stationary bar that is lower in intensity at 60-degree angle to the fly, we can simulate the response of the ellipsoid body. We find that the ellipsoid body activity pattern can still track the location of the input pattern in this scenario. We repeated the same process of model development and simulation for 2 other models of the central complex from literature, shown here in the middle and the right columns. These 3 concurrently published models each describe a different subset of the neurons in the central complex, as visualized by the corresponding neurons in the bottom row. In this video, we show 3 circuits side by side, showing the notebooks, the NeuroNLP morphology visualizations, and the circuit diagrams corresponding to these 3 different models. Note that for this specific circuit diagram, hovering over neurons shows additional modeling information for the loaded executable model, which is a feature of the circuit diagram. Towards the end of this video, we also show the simulation results, and let me fast forward to that section. And as you can see, by running some scripts, we can generate these visualizations. Employing the methodology I have just shown and using the same input to all 3 models, we performed a comparative study of their input and output behaviors. We see that although there are some slight differences in their response patterns, all 3 models show the tracking behavior of the ellipsoid body quite well. We then looked at the shared circuit features that these models all capture that allowed us to pinpoint the key circuit features giving rise to this behavior. We encourage you to read our preprint for these details. Similarly, we can use FlyBrainLab to compare models of the same species across developmental stages or datasets to shed light on the scaling principles. Here, for our second case study, we have looked at the early olfactory system of the fruit fly in 3 different datasets and used connectivity patterns between the cell types to construct similar but slightly different executable circuits. The early olfactory system of Drosophila consists of a number of parallel subcircuits whose receptor neurons express different olfactory receptors. Olfactory system of Drosophila is intensely studied, and we know a lot about the cells forming the early olfactory systems for both adult and larval Drosophila. Here, I am showing the neurons retrieved using FlyBrainLab queries from 2 different datasets we have. We have integrated known facts about the circuit and our observations from the query results to create executable models. Our models suggest that the activity of the so-called projection neurons downstream of the olfactory sensory neurons in each of these subcircuits are normalized in some sense, and encode the relative affinity of their olfactory receptor and the delivered odorant to the affinities of all other olfactory receptors in both adult and larval flies. To be more clear, we find that for both of these circuits, despite their differences, the projection neuron, or PN, responses are more similar to the affinity, which is in some sense the input to olfactory sensory neurons, rather than the responses of the olfactory sensory neurons themselves, whose activity patterns look different. This is interesting as PNs receive olfactory inputs from OSNs directly. All of these model development, analysis, and comparison examples we have went over, whether it's between models from different publications using the same dataset, or between datasets, or even between developmental stages, are significantly accelerated by the integration brought about by the FlyBrainLab platform. Recently, we have used FlyBrainLab as a tool for education on computational neuroscience in the Computing with Brain Circuits of Model Organisms course offered in Columbia University. Thank you for listening to our talk. I want to conclude by stating that in this age of widespread open data releases, there is a big opportunity in computational neuroscience for exploring, mapping, and understanding neural circuits of the fruit fly brain on a level of detail that will not be possible for other organisms for years. If you are interested to learn more about our work, you can read our preprints, download FlyBrainLab code on GitHub, explore our website, or follow us on Twitter. And again, thanks for listening to our